Welcome to the deep dive. Today we're tackling something, well, monumental. We are doing a rapid but really comprehensive immersion into the entire technological toolkit that built modern cell and molecular biology.
It's a huge topic.
It is. So if you're prepping for a big exam or you just feel totally overwhelmed by all the acronyms, FRAP, CRISPR, you are in exactly the right place. We're basically cracking open the entire lab notebook.
We are. We're going to synthesize the core principles behind every major technique, from the basic light microscope all the way up to cutting-edge genome editing.
And this deep dive is really designed to be your structured shortcut. We want you to understand exactly how biologists get the data that defines the entire field.
And it's so essential because cell biology, maybe more than any other science, is fundamentally defined by its technology. Our understanding of the cell, it didn't just happen. It was completely enabled by the invention of these incredibly sophisticated instruments.
So the tools aren't just accessories.
Not at all. They are the fundamental source of pretty much all the molecular knowledge we have.
And as soon as we start talking about tools that let us manipulate life at its absolute core at the genomic level, we have to talk about the responsibility that comes with that power. So our discussion really has to start with the ethical context, starting with the most powerful genome editing tool we have today.
And that, of course, is the CRISPR-Cas9 system. It's just the perfect illustration of the speed and, frankly, the power of modern biotech. To get the mechanism, you have to visualize the core parts. You've got this large Cas9 protein, it's an enzyme, and it's physically attached to something called a guide RNA.
So the RNA is the guide, like the name.
It's the guide. The whole complex acts like a highly targeted surveillance system.
Right.
It scans along all the DNA in the genome until that guide RNA finds a sequence that matches it perfectly.
And when it finds the match.
Once the match is found, Cas9 does its job. It's basically a pair of molecular scissors, and it just cleaves the DNA at that precise programmable spot. And that capability, that specific programmable DNA cutting, is what totally revolutionized the field, and at the same time, completely terrified the ethicists.
Yeah, that power, it immediately forced the scientific community to kind of pump the brakes decades ago. You can trace this all the way back to 1974, right after the first big breakthroughs in recombinant DNA.
Right, the ability to literally cut and paste genetic material from totally different organisms.
Even different kingdoms.
Exactly. And the concerns about the risks. I mean, what if you created a new pathogen by, say, combining a viral genome with some antibiotic resistance genes? The concerns were so great that a group of researchers, led by Paul Berg at Stanford, called for a voluntary international moratorium on all of it.
Just stop everything.
Stop everything. And this really extraordinary step, it led to the Asalomar Conference in 1975. They brought together 140 of the world's leading molecular biologists, plus lawyers, government officials, journalists, all to just hash it out.
Wow.
And what came out of it was monumental. They agreed to lift the moratorium so research could continue, but, and this is a huge, but only within a really strict set of defined restrictions, which the NIH adopted the next year. It set this crucial precedent.
That scientists have to regulate themselves.
They have to proactively guide the use of their most powerful tools.
Yes. And that history is so relevant today, especially with genome editing. In 2015, you had Jennifer Doudna, one of the pioneers of CRISPR, calling for another global ban, this time on modifying human germline cells.
The cells whose genetic changes could be passed down to all future generations.
Right.
The stakes couldn't be higher.
And this caution was then followed by the first big international summit on the topic. But then the abstract danger became horrifyingly real in late 2018. That's when He Jiankui, a Chinese scientist, made that shocking announcement that he'd supervised the birth of 2 CRISPR-edited human babies.
The goal being to make them resistant to HIV. The global reaction was immediate and just overwhelmingly negative. He was condemned by everyone, he faced legal consequences, and it led to this renewed, urgent call for another global moratorium.
And Paul Berg, the guy who organized the original Asalomar conference, he endorsed this new call, too.
He did, over 4 decades later. The core issue is still the same. How do you globally regulate a technology this powerful and, frankly, this easy to deploy?
It's a profound thought. The technologies we're about to talk about don't just generate knowledge. They put this enormous ethical weight right on the shoulders of the researchers. So with that critical context set, let's shift to the absolute bedrock of cell discovery, visualization. We'll start with section 1. Visualizing the cell, beginning with the very first tool, the light microscope.
The light microscope is where it all began. It's how we discovered cells even exist. And when we talk about its components, we're talking about the compound light microscope. So light comes from a source at the bottom.
Yep.
It travels up, and it hits the substage condenser lens.
And the condenser's job is really critical, right? It has to take all those diffuse rays of light and focus them into this tight, intense cone that actually illuminates the specimen.
Precisely. Once the light hits the specimen, the objective lens, that's the one right above the slide, collects the rays. And those rays have now diverged into two important paths.
Okay.
You've got the bright background light that the specimen didn't alter, and then you have the light rays that were altered, that were diffracted by the structures in the cell.
And the objective lens forms an image from those.
It forms a real, enlarged image inside the microscope column. And that image then becomes the object for the final lens system, the ocular lens, or the eyepiece.
Which produces the final image that actually hits your eye. And the total magnification is just, you multiply the objective lens power by the ocular lens power. Simple math.
Simple math. But magnification, as we all know, is only half the story. We have to talk about resolution.
Right. Resolution is the real limit. It's the ability to see two separate points as two separate points instead of just one big blur.
Exactly. Think about looking at densely packed chromatin. If the strands are too close, they just blur into a single mass no matter how much you magnify it. You can switch to a higher power eyepiece and the image gets bigger, but eventually you hit a point
where you're not gaining any new detail.
You're gaining no new detail. That is the definition of empty magnification. The image size goes up, but the quality, the fine detail, actually gets worse because you've already exceeded the objective lens' ability to capture that detail. The quality of a scope is all about its resolving power.
And there's a hard physical limit to this, governed by the physics of light itself, by diffraction.
That's right. Even a perfect tiny point in the specimen gets rendered as a small disk in the image. And if two of those points are so close that their disks overlap too much, you just can't distinguish them.
And that's where the resolution equation comes in.
It is. So D, which is the minimum distance you can resolve, equals 0.61 times the wavelength of light lambda, divided by a term called n sine alpha.
And that denominator, n sine alpha, that's the numerical aperture, or NA, it's a number stamped right on the side of the lens, and it tells you how much light it can gather.
Right. And for a lens looking through air, the refractive index, n, is just 1.0. So your max NA is also 1.0. But to do better, we use oil immersion.
You put a drop of oil between the lens and the slide.
You do. And that oil has a higher refractive index, about 1.5, which lets the lens gather more light. So you can get a practical NA up to about 1.5.
Okay, so if you plug in the shortest possible wavelength of visible light and that maximum NA you can get with oil, the math works out to a theoretical limit of resolution for a light microscope of just under 0.2 micrometers.
Or 200 nanometers.
200 nanometers.
And that 200 nanometer limit is an iron ceiling. It's good enough to see big organelles, the nucleus, mitochondria, sure, but it is nowhere near what you need to see ribosomes or microtubules or individual proteins. This limit is what drove the development of every other microscopy technique we're going to talk about.
Okay, so moving from the theory to the practice of actually seeing something, you have the problem of visibility, which is really about contrast.
Exactly. An unstained cell is almost totally transparent. It's like dropping a clear glass bead into immersion oil that has the same refractive index. It would just vanish. Visibility depends on the difference in appearance between an object and its background.
So the most basic way to boost that is with a bright field microscope, where the background is bright. But to see any real detail, you have to do a lot of prep work on the specimen.
And that prep it's harsh. It involves killing and modifying the cell. First, you have chemical fixation, using something like formaldehyde to immobilize everything, then dehydration and alcohol, and finally embedding it in paraffin wax so you can slice it thin.
And then staining. You need dyes to make things visible, like the Fulgen stain, which is specific for DNA, but the whole process is destructive. It kills the cell. So it's useless if you want to see anything dynamic.
Anything alive, yeah. Which brings us to the breakthrough that let researchers actually watch cells in motion: phase contrast and differential interference contrast, or DIC.
Okay, so how do these work?
Well, the phase contrast microscope makes transparent, unstained things visible by exploiting these really subtle differences in refractive index. Different organelles have different molecular compositions, so they bend light just a little bit differently.
And the microscope can detect that tiny difference.
It converts those invisible differences in refractive index into visible differences in intensity, so brightness and darkness. The genius of it is that the scope physically separates the direct light from the light that was diffracted by the specimen.
And then it recombines them.
It recombines them and causes them to interfere with each other. Where they interfere constructively, the image is bright. Destructively, it's dark.
So this was perfect for watching living cells.
It was a game changer. You could suddenly track mitochondria moving or watch chromosomes during mitosis. It gave us the first real movies of life inside the cell.
And DIC, or Nomarsky, that's a refinement on this.
It is. DIC gives you an image with this really distinct, almost three-dimensional quality, because the contrast depends on the rate of change of the refractive index across the specimen. It makes edges and boundaries pop with this crisp, shadowed detail.
So the next big leap was fluorescence microscopy. This seems to be where things get really powerful, turning the microscope into a tool for watching molecular dynamics.
It really was transformative.
Yeah.
The whole mechanism relies on fluorophores molecules that absorb photons of a specific short wavelength and then immediately re-emit that energy as light at a longer, less energetic wavelength. That's fluorescence.
And the microscope is built to handle that.
It is. It has special filters. One filter blocks everything from the light source except the specific excitation wavelength for your fluorophore. That pure light hits the specimen, excites the molecules, and the resulting longer wavelength emission light is what's captured to form the image.
The applications for this seem endless.
They're huge. The most common is immunofluorescence. You link a fluorophore to an antibody to pinpoint exactly where a specific protein is inside a cell. You can also use dyes that bind to DNA, or special dyes that measure things like calcium ion concentration.
But the real game changer here was green fluorescent protein, GFP.
Oh, absolutely. Discovered by Osamu Shiomamura in jellyfish. What made GFP so revolutionary is that it's a protein. It doesn't need an external dye. Its light-emitting part, the chromophore, forms automatically from its own amino acids.
So once the gene for GFP was cloned,
you could use recombinant DNA to just fuse the GFP gene sequence directly to the gene of any protein you were interested in. The cell then makes a chimeric protein, your protein, which is now literally glowing green.
So you can watch it move around in a living cell without killing it.
In a live, fully functioning cell. And thanks to Roger Tsien, we now have a whole rainbow of them, blue, cyan, yellow, and red variants from corals.
Which lets you track multiple things at once. I remember seeing that image of neurons in mice competing, with one expressing yellow and the other cyan. You could literally watch one win and the other lose in real time.
That's a classic example. Another beautiful one was visualizing the Golgi. They labeled an early Golgi protein in green and a late Golgi protein in red. And you could watch a single stack, a single cisterna, and see its color literally change from green to red over about 13 minutes.
Direct visual evidence for the cisternal maturation model.
Exactly.
And the precision gets even finer, down to measuring molecular proximity with FRET.
FRET, yeah. Fluorescence Resonance Energy Transfer. It's basically a nanoscale ruler, super effective in that tiny 1 to 10 nanometer range.
So it needs two fluorophores, a donor, and an acceptor.
It does. And the trick is that the excitation energy only transfers from the donor to the acceptor if they're spherically incredibly closer than 10 nanometers. If the transfer happens, the donor's fluorescence goes down and the acceptor's goes up.
So you can ask questions like, is this protein changing its shape?
Precisely. For the PKG signaling molecule, when cGMP binds, it causes this conformational change that brings the donor and acceptor fluorophores close enough for FRET to occur, and you detect that as a change in the light ratio.
And then there's FRAP, which asks a totally different question.
A different question. FRAP fluorescence recovery after photobleaching asks, is this protein moving and how fast?
The idea is you bleach an area with a laser.
Right. You have a population of fluorescently labeled proteins like GFP tubulin. You hit a tiny spot with an intense laser pulse, which irreversibly bleaches those specific molecules. Then you just watch.
And you time how long it takes for new unbleached proteins to move into that dark spot.
And that rate of recovery tells you about the protein's mobility, diffusion rates, or the dynamic instability of something like a microtubule. It's incredibly powerful.
All these techniques, though, they still had that problem with out-of-focus light blurring the image, which was finally solved by laser scanning confocal microscopy.
Invented by Marvin Minsky. And the problem he was solving was simple. In a regular scope, light from above and below your focal plane creates a haze. The confocal solution is just elegant.
It uses a laser to scan a single really thin plane of the specimen.
It does. It eliminates only that one optical section. But the real key, the magic trick, is that the light emitted from the fluorophores in that plane has to pass through a tiny pinhole aperture before it hits the detector.
And that pinhole is Confocal.
It's confocal with the illuminated plane, so only light rays coming directly from that plane can get through. Any scattered out-of-focus light from above or below is physically blocked by the pinhole.
And the result is a stunningly crisp image of an ultra-thin slice.
A slice as thin as 0.3 micrometers. And since they're digital images, you can just computationally stack hundreds of them to create a full, high-resolution 3D model of the whole thing.
But even with confocal, we were still stuck with that 200 nanometer diffraction limit until super-resolution fluorescence microscopy came along.
The Nobel-winning work of Betzig, Warner, and Hell. This really did break the barrier. A technique like storm stochastic optical reconstruction microscopy is just mind-bendingly clever.
It uses special fluorophores, right? Photoactivatable ones.
Yes, and here's the trick. You only activate a tiny random handful of them in each imaging cycle.
So only a few are glowing at any given moment.
Exactly. Each one that glows still looks like a blurry diffraction spot hundreds of nanometers wide. But because there are so few of them, you can use an algorithm to find the precise mathematical center of each spot with very high accuracy.
And then you just repeat that thousands of times.
You do. You build up the final image, one molecule at a time from all those precise coordinates, and you end up with an image with 10 to 20 nanometer resolution. The difference is just breathtaking. You can suddenly see individual microtubules that were just a blur before.
Okay, one last light technique. Light sheet fluorescence microscopy.
This is for imaging large, living things over long periods. The method illuminates the sample with a very thin, flat sheet of laser light, but, and this is key, it's oriented perpendicular to the observation lens,
So you're only lighting up the exact plane you're looking at.
which drastically reduces photo damage and photo bleaching. It lets you watch, say, an entire embryo develop over days. The newest versions, like lattice light sheet, are what enable true 4D imaging, 3 dimensions of space, plus time. It's incredible for watching development unfold.
So to break that 200 nanometer barrier entirely, we have to abandon light and turn to electrons. This brings us to the transmission electron microscope, or TEM.
And the TEM gives you a 100 to 200-fold increase in useful resolution over a light microscope. Suddenly you can see the internal cristae of mitochondria, the fine structure of muscle fibrils, things that were completely invisible before.
And this is possible because the resolution limit is tied to wavelength.
Directly. And electrons have wave properties. Their wavelength depends on their speed, which we control with accelerating voltage. At say 60,000 volts, an electron's wavelength is just incredibly short, 0.05 angstroms.
Which should give you insane resolution.
Theoretically, yes. But practically, the magnetic lenses in a TEM have severe spherical aberration. So that forces us to use a very small numerical aperture, which brings the practical resolution for looking at a cell down to about 10 to 15 angstroms. Still, vastly better than light.
And the machine itself is this huge, tall column.
A vacuum column, yeah. Because air molecules would just scatter the electrons. At the top, you have a heated tungsten filament. That's the electron source. They're accelerated by high voltage and focused by electromagnetic lenses.
The electrons pass through the specimen, hence transmission.
Exactly.
Yeah.
The ones that make it through strike a phosphorescent screen at the bottom, creating the image. Though now it's almost all digital CCD sensors.
And the contrast, the image itself, comes from how the electrons are scattered.
It does. Biological material, carbon, hydrogen, oxygen, is really bad at scattering electrons. It's basically transparent. So you have to stain the tissues with heavy metals, like uranium and lead.
Because heavy metals are good at scattering electrons.
Very good. So the dark regions in the final image are where a lot of electrons got scattered and lost. The bright regions are where electrons passed through easily because there was less stain.
And the specimen prep for TEM is notoriously difficult.
It's an art form. You need perfect fixation with glutaraldehyde and osmium tetroxide, dehydration, embedding in rock-hard epoxy resins, and then the sectioning. The sections have to be ultra-thin, less than 0.1 micrometers thick.
That's about the thickness of 4 ribosomes stacked on top of each other.
It's unbelievably thin.
Yeah.
And you have to cut it with a diamond knife, then pick it up on a tiny metal grate and stain it again with heavy metals.
Because all that chemical prep can create artifacts, there's a better way. Cryofixation.
A much better way. You freeze the sample so fast, instantaneously, that the water doesn't have time to form destructive ice crystals. It becomes this glass-like, non-crystalline solid called vitrified ice.
And that's what's used in cryo-electron tomography, or cryo-ET.
Right. You take one of these flash-frozen, unfixed, fully hydrated cells, and inside the TEM, you tilt it at many different angles, capturing a 2D image at each tilt.
Then a computer puts it all together.
A computer aligns and merges all those 2D images to create a full 3D reconstruction, a tomogram. It's an amazing technique because it bridges the gap between the cell level and the molecular level. You can see the 3D arrangement of things like polysomes inside the cell.
The EM is also great for looking at small, isolated particles, using techniques like negative staining.
With negative staining, you surround the particle, like a virus or a ribosome, with a heavy metal stain. The stain doesn't penetrate the particle, so the particle itself appears bright against a dark stained background. It's a great way to see overall shape.
And shadow casting gives a 3D effect.
It does. You put the specimen in a vacuum and evaporate a heavy metal, like platinum, from an angle. It coats the surfaces facing the source, leaving a shadow behind the particle. Those shadow areas appear bright, giving you a really nice sense of relief and dimension.
Okay, let's shift to looking at surfaces. This brings us to freeze fracture replication.
This is a really powerful and unique way to see the inside of cellular membranes. You rapidly freeze the tissue, and then you crack it with a knife edge in a vacuum chamber.
And the crack follows a specific path.
It does. The fracture plane loves to travel through the hydrophobic core of membranes, splitting the lipid bilayer right down the middle.
So you're looking at the internal face of the membrane.
Exactly. You then coat that fractured surface with heavy metal and carbon to create a durable replica. This technique was absolutely fundamental. It gave us the first direct visual evidence of integral membrane proteins embedded in the lipid bilayer, which was crucial for validating the fluid mosaic model.
And there's a variation called freeze etching.
Yeah, that's where you sublimate a thin layer of the surface ice before you make the replica. It can reveal external surfaces and deeper structures in really high relief.
Now, a completely different tool for surfaces is the Scanning Electron Microscope, or SEM.
Fundamentally different from TEM. The SEM's whole purpose is to examine the surface of an object. It's what gives us those iconic 3D-looking images of everything from insect heads to viruses.
And the prep is different, too. You have to get all the water out without the sample collapsing.
Right, usually through something called critical point drying. Then you coat the whole thing with a thin layer of metal, like gold, to make it conductive.
And the image isn't formed by electrons passing through.
No, it's formed by electrons that bounce off the surface, secondary electrons which hit a detector. A beam scans across the surface, and the signal from the detector controls the brightness on a screen. So it's an indirect image that reflects the surface topography.
The SEM is famous for its incredible depth of focus.
It's hundreds of times greater than a light microscope, which is what gives you that spectacular 3D look.
And a more advanced version is FIB-SEM.
Focused Ion Beam SEM. This is really cool. It combines an ion beam, which is used to carefully shave off ultra-thin layers of the material, just a few nanometers thick.
With the regular electron beam for imaging.
Right. So you shave off a layer, take a picture, shave off another layer, take another picture, and then you computationally stack all those 2D images to build a super high-detail 3D reconstruction of the internal structure. It's amazing.
Finally, in this section, we have the Atomic Force Microscope, or AFM. This doesn't use light or electrons at all.
No, it's a scanning probe instrument. It has this razor-sharp tip that it scans over the specimen's surface, and it monitors tiny changes in the oscillation of the cantilever holding that tip and converts that information into an incredibly detailed 3D topographic map.
The big advantage here is that it can image a single molecule.
A single individual molecule.
Mm-hmm.
Unlike X-ray or cryo-EM, which give you an average structure from millions of molecules.
Right.
And even better, high-speed AFM can take rapid sequential images, like 7 frames per second.
So you can actually watch molecules in action.
You can literally get a movie. You can watch a single myosin V molecule walking along an actin filament. And you can also use the probe as a nanomanipulator to stretch things or measure the binding force between a ligand and its receptor. It's an amazing tool.
Okay, let's move beyond visualization. Section 2 is about tracers, culture, and fractionation. We'll start with radioisotopes.
A tracer is just any substance you can easily monitor as it moves through a system. And radioactively labeled molecules are perfect because they behave exactly like their normal, non-radioactive counterparts in chemical reactions.
And the basis of this is isotope.
Right. Same number of protons, so it's the same element, but a different number of neutrons. Radioisotopes, like tritium, are unstable and they spontaneously disintegrate, releasing radiation that we can detect.
And their instability is measured by their half-life.
It is. I mean, early researchers used carbon-11 with a half-life of only 20 minutes. Experiments were literally a race against the clock. The discovery of carbon-14 with a half-life of 5,700 years was a huge step forward.
So what are the common isotopes we use?
Well, phosphorus-32 is great for nucleic acids, and sulfur-35 is common for proteins. And we detect them in two main ways.
Okay.
The first is liquid scintillation spectrometry. You mix your sample with phosphors, which absorb the energy from the decay, and emit it as a pulse of light. A photomultiplier tube then quantifies that light, giving you a precise number for how much radioactivity is there.
And the second method is for finding where the isotope is.
Exactly. That's autoradiography. The radiation activates a photographic emulsion, just like X-rays do. When you develop it, you see tiny silver grains right over the spot where the isotope was located, say, within a tissue section.
Next up, cell culture.
Mhm.
This seems like one of the most fundamental tools in all of modern biology.
It really is. Having these simplified, controlled in vitro systems is essential. You can get large quantities of a single cell type, you can study complex activities, and you can control the environment with incredible precision, adding specific drugs or hormones.
The media the cells grow in used to be pretty soupy.
Oh yeah, lots of serum and embryo homogenates. The goal now is a completely defined, serum-free medium with only purified, known growth factors. And, of course, because it's so nutrient-rich, you need absolute, strict sterility.
And we distinguish between primary culture, which comes right from an organism.
And secondary culture.
Hmm.
Normal cells have a finite lifespan.
Yeah.
They undergo senescence after maybe 50 to 100 divisions. So most research actually relies on cell lines.
Like the famous helo cells.
Right. These are cells that have undergone genetic modifications that make them immortal. They'll just grow indefinitely, which is obviously very useful.
There's a big push now for 3D culture, moving away from growing cells on flat plastic.
A huge push, because cells grown on a flat 2D surface behave unnaturally. When you grow them in a 3D matrix that mimics the extracellular environment, they adopt their proper shape, and their signaling and differentiation patterns are much more realistic.
And this is essential for growing things like organoids.
Exactly, these miniature, self-organized, organ-like structures. They're much better disease models than a flat layer of cells.
An innovative technique here is microfluidics, the lab on a chip.
These are amazing. Devices with micron-scale channels that give you incredibly precise control over tiny volumes of fluid. They're perfect for high-throughput screening of single cells and for integrating multiple steps, imaging, fixing, staining all on one small device.
So if we want to study the biochemistry of these cell parts, we need to get them out in bulk. This brings us to fractionation of a cell's contents by differential centrifugation.
Right. The principle is just separating organelles based on their size and shape using centrifugal force. The first step is homogenization, breaking the cells open in an isotonic buffered solution, like sucrose, to keep the organelles from bursting.
Then you spin them in a centrifuge.
You do, in a series of steps at increasing speeds. A low-speed spin pellets the biggest things, nuclei and whole cells. A higher speed brings down mitochondria, lysosomes, and peroxisomes. And then you need an ultracentrifuge, with forces up to 500,000 times gravity, to pellet the really small stuff like microsomes and ribosomes.
But those first fractions aren't very pure.
Not at all. They're crude. So for further purification, you often use density gradient equilibrium centrifugation. You layer your crude fraction on top of a density gradient, and the organelles sediment down until they reach a point that exactly matches their own buoyant density, forming these nice, distinct, pure bands.
And these purified fractions are the raw materials for cell-free systems.
Exactly. They let you study complex processes like protein synthesis or DNA replication outside the chaotic environment of the living cell, using just the components you need. It's a hugely powerful approach.
Okay, on to section 3, all about proteins, isolation, purification, and fractionation. This is a huge challenge, right? A cell has thousands of different proteins.
And many of them are present in incredibly tiny amounts. So the measure of success, your metric, is the increase in specific activity. That's the ratio of your target protein to the total amount of protein. You need a good assay to track it at every step.
The workhorse tool for separation here is chromatography.
It is. The basic idea is that proteins separate based on how they partition between a mobile solvent phase and a stationary, immobile, porous matrix.
Let's run through the 3 main types. First, ion exchange chromatography.
This separates proteins based on their overall net ionic charge, which, remember, is very dependent on the pH of the solution. The matrix is either positively charged, an anion exchanger that binds negative molecules, or negatively charged, a cation exchanger.
And you get the proteins to come off the column by adding salt.
Right. You elute them by increasing the salt concentration, so the salt ions compete for the binding sites, or by changing the pH to neutralize the protein's charge.
Okay, second type, gel filtration chromatography.
Sometimes called size exclusion. This separates purely based on size, on hydrodynamic radius. The column is packed with porous beads.
And this is the one that seems counterintuitive. The big molecules come out first.
They do, because they're too big to enter the pores in the beads. So they took a direct path right through the column and elute quickly. The smaller molecules can diffuse into the beads, which slows them down, so they elute much later.
And third, the most powerful one, affinity chromatography.
This one is just beautiful. It exploits a protein's unique, specific biological interaction. An enzyme and its substrate, a receptor and its ligand, an antibody and its antigen.
So you attach the binding partner to the matrix.
You do. When you pass your cell extract over the column, only your target protein sticks. Everything else just washes right through. It can often give you near total purification in a single step. It's incredibly efficient.
Once you have a protein, you want to know what it interacts with. There's a screening tool for that called the yeast-2 hybrid system.
The Y2H system. It's a brilliant genetic trick for finding protein partnerships on a massive scale. It relies on rebuilding a functional transcription factor inside a yeast cell.
So a transcription factor has two parts, a DNA binding domain and an activation domain.
Right. And you create two hybrid proteins. One links the DNA binding domain to your bait protein. The other links the activation domain to a whole library of unknown fish proteins.
And neither one can work on its own.
Neither can. But if your bait protein and one of the fish proteins physically interact inside the yeast cell, they bring the DNA binding domain and the activation domain together. That reconstitutes a functional transcription factor, which turns on a reporter gene.
Which gives you a color change or something easy to detect.
Exactly. It's a signal that those two proteins are partners.
Now, for just separating proteins to look at them, we use Polyacrylamide Gel Electrophoresis, or PAGE.
Right. You use an electric field to pull charged molecules through a polyacrylamide gel, which acts as a molecular sieve. Separation depends on charge, size, and shape.
And when you're done, you can transfer the proteins from the gel to a membrane, which is called a blot.
It is. And if you want to find one specific protein in that whole mess, you do a Western blot, where you probe the membrane with a specific antibody against your target.
The problem with standard PAGE is that shape and charge can mess up the separation, which is why everyone uses SDS-PAGE.
Right. SDS is a detergent that unfolds all the proteins into a similar rod-like shape and coats them with a uniform negative charge that's proportional to their mass.
So since the charge and shape are now basically the same for everything.
Separation depends only on molecular mass. It lets you accurately determine the size of your protein.
And for separating thousands of proteins at once, there's two-dimensional gel electrophoresis.
This is a powerhouse technique. You separate the proteins in two steps. First, in one dimension, you separate them by their isoelectric point using isoelectric focusing. Then you take that gel, lay it on top of a second SDS-PAGE gel, and separate them in the second dimension by mass. You can resolve thousands of individual protein spots on a single gel.
Finally, for identifying what those spots are, you need mass spectrometry, or MS.
MS is the workhorse of proteomics. It measures the mass-to-charge ratio of gaseous ions. You digest your protein into smaller peptides, ionize them, and send them into the spectrometer.
And tandem MS, or MSMS, takes it a step further.
It does. It actually fragments the peptides inside the machine and measures the mass of those fragments. From that fragmentation pattern, you can determine the exact amino acid sequence of the peptide. This allows for rapid identification of hundreds of unknown proteins and, really importantly, their post-translational modifications.
Now, what about determining the 3D structure of a protein?
The classic method is X-ray crystallography. This requires you to grow a highly ordered, perfect crystal of your protein. You shoot X-rays at it, and the way they diffract off the electrons gives you a pattern that you can mathematically analyze to determine the electron density map, and from that, the atomic structure.
But getting that crystal is the hard part.
It's the bottleneck. Many proteins, especially membrane proteins, just refuse to crystallize. And that's why electron microscopy, or cryo-EM, has become so dominant.
With cryo-EM, you don't need a crystal.
No crystal. You just flash freeze the particles in a thin layer of vitrified ice and take thousands of 2D images of them in the EM, all in different random orientations.
And then computers do the heavy lifting.
Powerful computers average all those 2D images to generate a high-resolution 3D reconstruction. It's fantastic for large, complex, or flexible structures that you could never crystallize, like the ribosome.
Okay, let's dive into Section 4, Nucleic Acid Manipulation and Genomics. We start with fractionating nucleic acids.
And again, gel electrophoresis is key. Separation is based on length. Polyacrylamide for small pieces, egg rose gels for larger DNA fragments.
And for really, really big DNA, like whole chromosomes.
For that, you need pulsed field electrophoresis. This technique periodically changes the direction of the electric field, which forces those huge molecules to reorient themselves, and that allows for better separation by size.
Next, nucleic acid hybridization. This seems to be a core principle for almost everything in genomics.
It is. The principle is simple and powerful. Two single-stranded nucleic acid molecules with complementary sequences will spontaneously form a stable double-stranded hybrid. So you can use a labeled single-strand probe to find its matching sequence in a complex mixture.
And this is the basis of blotting techniques.
The basis. The southern blot is for DNA. You separate DNA fragments on a gel, denature them to single strands, transfer them to a membrane, and then probe that membrane with your labeled sequence.
And the northern blot is the same thing, but for RNA.
Exactly. It's a way to identify and quantify specific RNA molecules, which tells you about gene expression levels.
Moving on to actually making DNA, there's recombinant DNA technology. This starts with restriction enzymes.
These are the molecular scissors. They are enzymes from bacteria that recognize short, specific palindromic DNA sequences and cut the DNA backbone right at that spot.
And they can make sticky ends.
They often make staggered cuts, which produces these short, single-stranded overhangs that are complementary. These sticky ends are incredibly useful for pasting DNA fragments together.
So to make a recombinant DNA molecule, you cut DNA from two different sources with the same restriction enzyme.
Right, so they have matching sticky ends. You mix them together, the sticky ends anneal, and then you use an enzyme called DNA ligase to seal the backbones covalently. You've now made a new recombinant molecule.
And to make lots of copies of it, you need DNA cloning.
Right, you need to amplify it. You insert your recombinant DNA into a vector, usually a plasmid or a phage, and then introduce that into a host cell, like E. coli.
The plasmid has an antibiotic resistance gene on it, which is for selection.
It is, so you grow the bacteria on a plate with the antibiotic, and only the cells that successfully took up the plasmid can survive and form a colony. Each colony is a clone, containing millions of copies of your gene.
But the ultimate amplification tool is PCR, the Polymerase Chain reaction.
Invented by Kary Mullis. It's an amazing technique. It requires a heat-stable DNA polymerase, like PAC polymerase, which comes from a bacterium that lives in hot springs.
And the process is a cycle of 3 temperature steps.
3 steps, repeated over and over. First, denaturation. You heat the sample to about 95 degrees Celsius to separate the two DNA strands.
Second, annealing. You cool it down so that short synthetic primers can bind to the ends of the specific region you want to amplify.
And third, extension. You heat it to about 72 degrees, the optimal temperature for Taq polymerase, and it synthesizes new DNA strands starting from the primers. Each cycle doubles the amount of your target DNA. It's exponential amplification.
So from a single molecule, you can get billions of copies in a few hours.
It's incredible, and it's what makes things like forensics and DNA sequencing possible from tiny samples.
Speaking of sequencing, that's our next topic. The modern method is based on the Sanger method, right?
The Sanger-Coulson method, yeah. It was merged with PCR automation to give us cycle sequencing. This uses special modified nucleotides called DDNTPs, or dideoxyribonucleotides.
And these are special because they're missing the 3' hydroxyl group.
That's the key. So when the polymerase incorporates a DDNTP, the chain can't be extended any further. Synthesis stops. Chain termination.
And each of the 4 DDNDPSA, T, C, and G is labeled with a different colored fluorescent dye.
Correct. So you run the reaction, and you end up with a collection of DNA fragments of all possible lengths, each one ending with a color-coded terminal base.
Then you separate them by size.
Using capillary gel electrophoresis, which is so sensitive it can distinguish fragments that differ by just one nucleotide, a laser reads the color of the final base on each fragment as it comes out, and that gives you the DNA sequence directly.
Since then, we've had massively parallel sequencing, the second-generation systems.
These were a huge leap. They got rid of the cloning and the electrophoresis. They immobilize billions of DNA molecules on a surface and then identify the nucleotides as they're incorporated by polymerases in real time, in parallel.
They're super fast and cheap, but they produce short reads.
Very short reads, yeah. Less than 100 bases. So they're great for resequencing a genome where you already have a reference map, but they're tough for assembling a brand new genome from scratch, which is why third-generation sequencers are focused on getting much longer reads from single molecules.
So, to manage all the sequence information, we create DNA libraries.
We do. A genomic library is made from the total DNA of an organism. It contains everything: coding sequences, non-coding, regulatory, all of it.
In contrast, a cDNA library is made only from the messenger RNA.
Right, so it only represents the genes that were actively being transcribed in that particular cell type at that particular time. And since it's made from mRNA, it has a huge advantage. The introns have already been spliced out.
This brings us to putting DNA into eukaryotic cells and even whole animals.
Gene transfer. You can use viruses, which is called transduction, or you can use chemical or electrical methods to get naked DNA into cells, called transfection or electroporation, or you could just physically inject the DNA with a tiny needle called microinjection.
And that's how you make transgenic animals.
That's one way. You inject your foreign gene, your transgene, into an embryo, and if it integrates into the chromosomes, you'll have an animal that carries that new gene. This is crucial for making animal models of human diseases.
And this leads us right to the ultimate manipulation: gene editing and silencing.
We're now in the era of reverse genetics. We know the sequence, the genotype, and we want to figure out the function, the phenotype. One way is RNA interference, or RNAi.
This uses small double-stranded RNAs to trick the cell into degrading a specific target mRNA.
Exactly. So you get a temporary reduction or knockdown of the corresponding protein. It's a great way to quickly screen for the function of thousands of genes at the cellular level.
But the most powerful tool is genome editing using engineered nucleases. This is where CRISPR comes in.
Right. All these systems, the older ZFNs and TALENs, and now CRISPR work by making a precise double-stranded break in the DNA at a location you choose. The cell then has to repair that break.
And that repair process is where you can make changes.
That's where the magic happens. The cell can either just stitch the ends back together, which is often messy and can knock out the gene, or if you provide a template, it can use that template to repair the break, allowing you to insert, delete, or change the sequence with incredible precision.
And CRISPR is the simplest and most efficient system for doing this.
By far. It uses that simple guide RNA to direct the Cas9 nuclease to any spot you want. Its ease of use is what has driven this absolute explosion in genome editing research.
Okay, we've reached our final section, Section 5, the use of antibodies.
Antibodies are just invaluable tools. Their defining feature is their astonishing specificity. They can distinguish between two proteins that differ by just a single amino acid.
And the gold standard is the monoclonal antibody.
Absolutely. Traditional methods give you a polyclonal mixture of different antibodies, but the Nobel-winning technique from Milstein and Kohler allows you to make monoclonal antibodies.
This is where they fuse two cells together.
It's so clever. They fused a normal antibody-producing lymphocyte, which provides the specificity, with a malignant myeloma cell, which provides immortality. The resulting hybrid cell, a hybridoma, can be cloned and grown indefinitely.
And since it's a clone from a single cell, it produces a single, pure, homogeneous species of antibody.
A monoclonal antibody. And they're used for everything: protein purification, Western blots, diagnostic tests like home pregnancy tests, and even as powerful therapeutic drugs.
We also use them for immunolocalization to see where proteins are in the cell.
Right. You can do direct immunofluorescence, where the antibody itself is linked to a fluorophore, but it's more common to do indirect immunofluorescence.
This is where you use a primary and a secondary antibody.
Right. You have an unlabeled primary antibody that binds to your target protein. Then you add a fluorescently labeled secondary antibody that is designed to bind to the primary antibody.
And you do this for signal amplification.
Exactly. Because several secondary antibodies can bind to each primary antibody, the resulting fluorescent signal is much, much brighter. It's a more sensitive technique.
That brings us to the end of our sprint through the modern biological toolkit. We've gone all the way from the physical limit of the light microscope at Braille 0.2 micrometers to the incredible precision of cryo-EM and CRISPR.
These tools, when you look at them all together, they allow us to study life from the level of atomic structure all the way up to the complexity of a whole developing organism. Let's try to distill this all down into 3 core takeaways for you.
Okay, takeaway number 1. The fundamental hurdle is always resolution. And resolution is limited by the wavelength of the particle you're using. To overcome the light barrier, you either had to use clever computational tricks like super resolution or switch to a particle with a much shorter wavelength, like electrons.
Takeaway number 2. Amplification is the absolute bedrock of genomics. Cloning, and especially PCR, were the tools that made everything that followed, sequencing, editing, mapping, possible on a practical scale. You have to be able to make more of what you want to study.
And third, the power of specificity is everything. Whether you're talking about the targeted recognition of a monoclonal antibody, the site-specific cutting of a restriction enzyme, or the RNA-guided precision of CRISPR-Cas9, the ability to target and analyze one specific molecule in a cell full of millions of others is the key to understanding the whole system.
We started this conversation with the ethical gravity of editing the human germline. And now that we've seen the technologies that let us synthesize entire genomes and edit them with single-base pair precision, it raises this really profound and immediate question. If we can now see, map, and change every single component of a cell, what fundamental definition of life or structure or function will be the next thing to be irrevocably challenged by the next generation of biological techniques?
That is a critical thought to ponder as you continue to study the cellular world. Thank you for joining us for this deep dive into the indispensable tools of cell and molecular biology.